Let's stop here. This was Watson and Crick who gave the double helix structure for DNA. So double helix model, base pairing between the two strands of polynucleotide chain. So in yesterday's class, we studied how one polynucleotide chain is formed. It is made by joining many nucleotides. Many nucleotides, they join by which type of bond? Phosphodiester bond. That is, one nucleotide will join the other nucleotide by phosphodiester bond, and the phosphate will bind to the nucleoside. What is nucleoside? Nucleoside is sugar and the nitrogenous base. To this nucleoside, the phosphate will bind by phosphoester bond. Which carbon it is joining? Fifth carbon. Phosphate will join to the fifth carbon. Base, nitrogenous base joins to the first carbon. Nitrogenous base joins to the first carbon. Then fifth carbon, phosphate is binding. Third carbon, that OH will come. Hydroxyl. Third carbon, hydroxyl will come. OH. Phosphate on fifth carbon. Fifth carbon phosphate is there. Third carbon OH is there. And on first carbon there is the nitrogenous base. The nitrogenous base joins the sugar by which type of bond? Glycosidic bonds. Okay, glycosidic bonds. So Erwin Chargaff ratios between adenine and thymine. Okay, Erwin Chargaff, he gave a rule that the ratio between adenine and thymine and guanine and cytosine are constant, n is equal to 1. The ratio of A upon T and G upon C, ratio you know, A upon 1, or A is to T or G is to C is always 1. Okay, ratio cannot be 0, no? Ratio cannot be 0. Base pairing confers a very unique property. There is a unique property for the base pairing. Now let's see what is this unique property. Complementary to each other. Now the two strands which they bind, no, they are complement to each other. Okay, the two strands which binds are complement to each other. Now why they are complement? Because A will bind with whom? A always joins T and G always joins to C. So if I give you one strand A, T, G, C, what will be the other strand?
T,
A, T, G, C.
T, C.
A, T, G, C. So what will be the other strand? First is correct. A will bind to T. T will bind to A. G will bind to C. C will bind to G. So the other strand will be T, A, C, G. So if you know the one strand, you can find what will be the other strand. If I give you one strand, you can find out what will be the other strand. That is called complement to each other. So what will happen if you know the sequence of one strand? You can find the sequence of the other strand. Now, that is not important. Only important is Chargaff's rule. Chargaff is very important. They directly ask question on explain Chargaff's rule. So we'll do it later again. Chargaff rule in detail we'll do again. Okay. So salient features of double helix structure which was given by Watson and Crick. Features. Two polynucleotide chains. There will be two polynucleotide chains joined to each other by hydrogen bonds. Okay. So there are three different types of bond in DNA molecule. The first bond is, which is the first bond? Glycosidic bond. Second we studied is phosphodiester bond. And the third bond is hydrogen bond. So between the bases, that is A and T, will have double bond, double hydrogen bond. G and C will have triple hydrogen bond. Okay. Anti-parallel polarity. What do you mean by polarity? We had done yesterday. No, we had given pole names. What is the pole names? Numbers we had given. 5 dash and 3 dash. Okay. They are anti-parallel polarity. They have anti-parallel polarity. So if this strand is starting from 5 to 3, the other strand which is complementary will start from here, it will be 5 and end with 3. Hydrogen bond. Adenine forms 2 hydrogen bonds with thymine and guanine is bonded with cytosine with 3 hydrogen bonds. 2 chains are coiled in a right hand fashion. They are coiled. They are coiled in right hand fashion. Helix is 3.4 nanometer. The helix length, no? If we start from here, it will come here. This will be one helix. Okay. If you start from here, this will be one helix because this and this should join. When you turn it, it should join. So that helix is 3.4 nanometer. You all know what is nanometer? 10 raised to minus 9. Okay. So in each helix, no, from here to here, there will be total of 10 base pairs or 10 nucleotides on one strand of each helix. Total there will be 20. 10 from on this, 10 on the other. Central dogma is molecular biology, which states that the genetic information flows from DNA. The genetic information will turn into RNA, and from RNA it will give rise to protein. So this is how the genetic information is converted. From DNA, whatever information is in your DNA will first get converted to RNA. Then from RNA, it will get converted to protein. For example, melanin protein. The melanin, which is the pigment. And for that protein, there will be one DNA in your body, which gets converted to mRNA. That will study later. Types of RNA is there. tRNA, mRNA, and rRNA, which we'll study later. But just for time being, you have to know DNA to RNA to protein. This entire process is also there for you. So this is the structure. So this, it starts from here and it ends here. Okay. So this is 34 Armstrong. Armstrong is 10 raised to minus 10. So it will be 3.4 nanometer. 34 Armstrong. So total number of bases 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Total number of bases, if you count, it comes to 10. If you start from here till here, it is 10. So what is the gap between each base or between two base pairs? What will be the gap?
10.
If the total is 34 Armstrong, so what will be the distance between two base pairs?
34 divided by 2.
34 divided by how many?
10.
Total 10 are there from here to here.
0.4.
So 3.4 Armstrong. This is minor group and this is major group. So this is the basic structure of DNA. Okay, now let's go to the next part. Packaging of DNA helix. What is the total length of DNA in a human? Total length of DNA in human. Okay. What is the length of helix? Helix is 34 Armstrong or 3.4 nanometer. Okay. It is 3.4 nanometer. There. How many bases are there in one helical turn? 10. There are 10 bases. Next. What is the distance between two bases?
3.4
Armstrong. Okay. So here it will come to 0.34 or 0.34 nanometer. Total number of base pairs in human is 6.6 into 10 raised to 9. Total number of base pairs in human is 6.6 into 10 raised to 9. So can you calculate now? So what will be the length? This is the total number of base pairs. Total number of base pairs into the length of what? Why helix? Length between two base pairs. Length between two base pairs. Total number of base pairs is 6.6 into 10 raised to 9, and the length between each pair is 3.4 0.34 nanometer. Length between two base pairs. So what will be the total length? You count the number of, let's say for example, there are 10 students standing. The gap between each student is 1 meter. So what will be the total length? 1 into 10. 1 meter into 10. So it will become 10 meter. Similarly, here there is the gap between two base pairs is 0.34. So approximately the length will be 6.6 into 10 raised to 9 base into 0.34 nanometer. Okay. So what you will get this answer as 6.6 into 10 raised to 9 into 0.34. Calculate. Easy it is. Just consider this 0.34 as 0.33 to take approximation. Yeah. Consider 0.33. Let's see what you get answer. Don't use calculator. Okay. There is no need of calculator here.
Can we cancel? We divide both of them, expensive unit one, we divide 2 unit.
Yeah. Okay. Show pencil, then also see. Let's see. 6.6 into 10 raised to 9 multiplied by 0.34 into 10 raised to minus 2. Yeah. So this 10 raised to 9 and this 10 raised to minus 9 goes. You know, no? Because it will become 10 raised to 9 plus minus 9. Law of indices. What is left is 6.6 into 0.34. 4 6 are 24, 4 6 are 24, 26. 3 6 are 18, 3 6 are 18, 19. 4, 6 plus 8, 4, 9, 10, 2. You get around 32 something. Sorry, 9 plus 10, 11, 12, 22. Then how you put point?
After 3 decimal.
After 3 decimal point. This is 2, and this is 1. So it will become 2.2 meter. Okay, because you have cancelled the nanometer. So it comes to 2.2 meter approximately.
Total length of DNA.
Total length of DNA. Now, how is such a long polymer packed in a cell? What is the size of the nucleus? Very, very small. And in that nucleus, you have 2.2 meter DNA. So how come this is possible? That we have to see. So this is how your DNA is packed. In class 10, if you see, if you remember, this was the image which was there in your first chapter, cell division. First, there is DNA. DNA joined to histone molecule to form nucleosome. Nucleosome will join to form chromatin fiber. Chromatin fiber will join to form chromosome. Okay. So there, this is a prokaryotic DNA, a bacterial DNA. In bacterial DNA, there are no histone molecules. In bacterial DNA, they don't have any histone. In prokaryotic, such as E. coli, though they don't have a defined nucleus, they don't have a defined nucleus. Huh? There is no nucleus. The DNA is not scattered throughout the cell. DNA is not scattered everywhere. It is at the center. DNA is not scattered throughout. DNA being negatively charged, okay, DNA being negatively charged is held with some protein that have positive charge in the region termed as nucleoid. So this region is the nucleoid. Got? So DNA is negatively charged. Remember this. DNA is negatively charged. The DNA in nucleoid is organized in large loops held by protein. They are like loops. Now this is important. In eukaryote, there is a protein called histone. Okay, this is very important compared to previous. They directly ask, explain, draw a neat diagram of a histone octamer. So in eukaryote, we have a positively charged basic protein called histone. Okay, now histones are rich in basic amino acid residues like lysine and arginine. Now, how this histone is getting positively charged? Because we have different types of amino acids. Some are positively charged amino acids. So this lysine and arginine, they are positively charged amino acid, and this they join to form histone protein. So this lysine and arginine, they join to form a protein. You know, amino acids join to form protein. Fatty acids join to form fats. Okay? Glucose joins to form carbohydrate. So this is positively charged. So histone is positively charged because it is rich in lysine and arginine. Histones are organized to form a unit of molecule called histone octamer. What is octamer?
8.
8. So 8 histone molecules will join together to form a octamer. The negatively charged DNA is wrapped on this octamer. Okay. Since DNA is negative charge, histone is positively charged, they will come close to each other. And this negatively charged DNA is wrapped around a positively charged histone octamer to form a structure called nucleosome. Okay. So you write nucleosome is equal to nucleosome. They'll copy from you. Since you are writing. Nucleosome is equal to histone plus DNA. Histone and DNA will join together to form a nucleosome. The negatively charged DNA is wrapped around the positively charged. Again, it is called as nucleosome. So this is the structure. And nucleosome, they will, I have told you already, nucleosome will join to form chromatin. Chromatin will join to form chromosome. So this is nucleosome structure. The proteins are H2A, H2A, 2 molecule of H2A, H2A, 2 molecules of H2B, H2B. Then there is H3 and H4, two molecules. What is H?
Histone.
Histone. H for histone. Histone 2A, histone 2B, histone 3 and histone 4. All multiplied by 2 will give you 8. So this is how it is formed. Okay, next. Now the last is, now packaging of chromatin at higher levels required additional set of protein which are collectively called as non-histone protein. Now histone is there. Already we have many histones together. Histone will join to form what? Chromatin. So they should also bind into each other or wound to each other, wound on each other. They will wound like a telephone wire. Okay. So for that, there are special sets of protein called non-histone chromosomal protein. A typical nucleus, in a typical nucleus, some region of chromatin are loosely packed. Okay, and so these are referred to as euchromatin. Okay. Some region of DNA loosely packed called as euchromatin, and the tightly packed chromatin is called as heterochromatin. The chromatin that is more densely packed and stains dark is called as heterochromatin. Euchromatin can be said is said to be transcriptionally active chromatin, whereas heterochromatin is inactive. Okay, euchromatin is active and heterochromatin is inactive. So I'll explain what is euchromatin and heterochromatin. What is central dogma? Central dogma is from DNA you get RNA, from RNA you get a protein. So euchromatin will give a protein, will undergo transcription and translation, which is there for you, will give you protein, but heterochromatin they will not give any protein. Okay, this is how it is bound. These are non-histone protein. This is histone, and they will join together. This is the last one.